Okay, we're going to start off with chapter 23. This involves several different, somewhat related topics: metabolism, nutrition, energetics. Now, this would not substitute for a full nutrition course, but it's to give you a taste and perhaps a better understanding of why. We just finished the digestive system, which was chapter 22. Why is it we spend so much time about this? Well, digestion is absolutely important, but the chemistry behind it will give you a much better understanding of why we consume what we consume. So, moving forward, we're going to first talk about metabolism. This is the sum of all catabolic and anabolic reactions. Now, if you get catabolic and anabolic confused, catabolism is the breakdown processes. Anabolism is the synthesis processes, in other words, the build up. You've known if it gets confusing for you, think of the term anabolic steroids. What do? They bulk somebody up. So they cause synthesis of muscle proteins to be initiated. Now, when we talk about the term nutrient pool, these are nutrients distributed in the blood, and they're available for anabolism or basically the catabolic process, which ultimately is involved with production of ATP. Okay. Now, when we talk about aerobic metabolism, this occurs in the mitochondria. We're going to talk about how these steps occur. As you can see here in this diagram, you have a variety of organic molecules: amino acids, lipids, simple sugars. Okay. When I say simple sugars, think of monosaccharides. Okay. Lipids, the best way to understand it is monoglycerides, fatty acids, things like that. When you talk about cholesterol is used more for the structure, either to make hormones, steroidal hormones, or it is involved in implementing and using it in the actual cell membranes. But we'll move forward on this. If you notice here, we have this catabolism, anabolism structure here. When we talk about anabolism, it's not just growth and secretion and nutrient reserves, but it's also involved with maintenance and repair. When we talk about getting energy out of this, from the nutrient pool, particularly with just glucose, you can get a little bit of ATP in the cytoplasm, but the bulk of ATP production, and keep this in mind, is really in the mitochondria. Now, cells use nutrients from the nutrient pool for metabolism. We've been talking about this and we'll continue to talk about it. The thing is that all basic cell activity really requires ATP. What you're doing is you're taking organic compounds and basically you're going to either do synthesis or you're going to break them down. Synthesis, you're going to make triglycerides, you're going to make glycogen. In other words, this is a situation where, okay, glycogen is used particularly in the liver and in the skeletal muscle when you take the simple sugars and make them into a complex polysaccharide. That's for energy storage. The proteins, well, we need proteins. Yes, you need proteins in bone, you need proteins in the skeletal muscles, you need them all over the place. But sometimes you have to understand that proteins can be broken down. Particularly, you're going to see this at the end of the chapter. The materials to be broken down will eventually yield ATP. That's the key point to keep in mind. Now, nutrients extracted from food in the digestive system are used by these various tissues. In liver cells, we end up taking the fatty acids, the monoglycerides, and we form triglycerides. Also, you will synthesize, in some cases, glycerol. You'll also take the monosaccharides and make glycogen. Now, here's a tricky little part to keep in mind. Neurons. Neurons do not take, they're fussy. If you've ever had a brother or sister who was a fussy eater, or you were a fussy eater, neurons are fussy. They only take glucose for their fuel. In starvation, there is a shunting off of breaking down of amino acids and breaking down of lipids and ketone bodies and all these other products, they can be used by things like skeletal muscles and other structures, so that we basically just use the glucose only for the neurons. Excuse me. Skeletal muscle cells, they can metabolize fatty acids. They can store glucose as glycogen. This is where we have a similarity in the skeletal muscles and in the liver cells, in that what they can do is take some of this excess glucose that occurs when you have feeding time and store it for subsequent use, and it's basically taking glucose and making it into glycogen. Enough said there. Adipocytes, these convert fatty acids to triglycerides, and in starvation, it's reversed. So they break down triglycerides and free up the fatty acids into the systemic circulation. Now, here we have the nutrient pool. You see fatty acids, glucose, amino acids. Yes, they can be made. Amino acids, the building blocks for proteins. Glucose would be the building blocks for basically making glycogen. Fatty acids can be used and stored as triglycerides. The other thing to keep in mind, though, is we can go in the reversal. Now, a lot of people don't always understand clearly. Amino acids can be broken down. They have to be deaminated first. We'll get into that in a little while. And depending on the structure of the individual amino acid, will either be put into around part of glycolysis or into the Krebs cycle, and you will yield energy, production of ATP. Fatty acids, what you do is you just basically picture sort of a long chain, and you chop off 2 carbons on that long chain of the fatty acids, and you throw them into the Krebs cycle, and you eventually get, yes, ATP production. Okay? So keep in mind that these are the anabolic pathways and the catabolic pathways. Notice that the nutrients are absorbed through digestion, and basically they're going to be shunted into the various types of cells. Adipocytes, skeletal muscle. Cells are most tissue. Nervous tissue, remember, glucose. Liver cells, we can do a lot of fun things with those. Okay, so let's talk about glycolysis first. It's the first step of glucose metabolism. Glycolysis is an aerobic process. We don't need oxygen. Now, if you remember in your medical microbiology, let's cross from one course to another. This is something you see consistently with certain organisms. But here's the main point. Glucose, which is a 6-carbon molecule, will be broken in half. You'll get 2 3-carbon molecules in the end. You get some pyruvate. That's what this 3-carbon molecule eventually is, pyruvate. In the process of taking 1 molecule of glucose and going through glycolysis, you get 2 net ATP produced. Now, you ever heard the saying, to make money, you need to spend money? Well, that's true, because in the process of this glycolysis, you're going to have to use 2 ATP molecules in the beginning. In the end, you get 4 ATP molecules, and I'm trying to help you to avoid getting confused here. So basically, 4 minus 2 gives you 2. So there's 2 net that are produced. You also have 2 NAD molecules that accept hydrogens from this process, and they form NADH. They will be used later on. Now, the pyruvate can be broken down further to acetyl groups, which are 2-carbon structures. This is occurring, though, inside of the mitochondria. Okay? Now, before you have a massive, oh my gosh, I'm not asking you to understand every one of these steps. If you were a biochem major, yes, you're not. So don't sweat it. Notice that you take the glucose, you begin to use a couple of ATPs because you're going to take off the phosphate from them. Eventually, you produce 2 here and 2 there, and basically 4 total produced minus the 2 that you initially had, and you had a net gain of 2 ATP molecules. Okay, real simple. Don't panic. I'm not asking you to go through all of these steps and the enzymes. Oh yes, and the biochem majors have to know the enzymes too. Haha, we don't. Okay, You can look it up in a book. The key point is that this is the first step in breaking down glucose, A. B, you get only 2 ATP molecules net out of it. You also make some water, yay. You also make 2 molecules of NADH, which is going to be used by the mitochondria to help produce more ATPs later. Now, oops, there we go. We're going to go to the next step. The next step is known as Krebs cycle, or the TCA cycle, or the tricarboxylic acid cycle, or the citric acid cycle. Krebs was the one that originally worked with, I believe it was mincing pigeon breast muscle. Pigeons, birds, and they have a lot of power that goes through this. That's how he was able to discover this, and I believe, yes, he did get a Nobel Prize for the work. This is all part of what we call aerobic metabolism. It is an oxygen-requiring process. Pyruvate is broken down to yield energy, and this is going to be a multi-step cyclic process. And what you have is the TCA cycle, or the citric acid cycle, is the first part of that step. Second step is electron transport chain, but all of this occurs in the mitochondria. So this brings us up to this important point. Why is it that we study about the mitochondria? Because this is the actual powerhouse of the cell, produce lots and lots and lots and lots of ATP, so cells can do all sorts of functions, okay? Now, pyruvate loses one of the carbons. That becomes part of CO2. There is a coenzyme called CoA that attaches to the acetyl group, and it starts going through this cyclic process. And in the end, also, NAD and FAD get hydrogens. Now, these hydrogens attached will eventually be used in the electron transport chain to produce, yes, more ATP. And if you look at the cycle, you do get actually a little bit of ATP made. Here it is down here. So you go down once, you go down twice, because you're going to have 2 acetyl groups, because you ended up with 2 pyruvate groups. But they all have to go inside of the, basically the mitochondria. Every time that you lose carbon, it becomes the carbon dioxide. And by the way, don't worry this much about the GTP, GDP. Yeah, it happens, but it gets converted very quickly into another molecule of ATP. This process strips out hydrogens, and as it does, it makes more of the NADH and FADH2. These will all play a key role in the electron transport chain, and that's where you get lots of ATP made. Here's another diagram of this. Yes, you do not, everybody say it, do not have to memorize all the steps. That's for the biochemists. What you do have to understand is that the citric acid cycle is absolutely essential. It's the first step of this aerobic metabolic process, okay? And it helps to break down the carbon so that you can yield a lot more energy. So let's talk about the electron transport chain, shall we? This is a process that establishes a proton gradient. In other words, across a membrane, you've got a lot of protons on one side, which is another way to say hydrogen ions. And then you've got very little on the other side. So what's going to happen? Well, if you go down the gradient, concentration gradient of these protons, you're going to be able to use that just like a dam holds up water, and you have the water go through these special tubes, sluices as they're called, and the turbines get turned and they produce electricity. In this case, we're not producing electricity, we're going to produce lots of ATP. This is a process that's called oxidative phosphorylation, the generation of ATP as a result of transfer of electrons from NADH and FADH2 to oxygen by a sequence of electron carriers within the mitochondria. We call this the electron transport chain because what happens is that you have a bunch of these integral and peripheral proteins on the inner membrane of the mitochondria. Remember, the mitochondria is unique. It has an outer membrane, an inner membrane. And literally, as you have these electrons bouncing around from one step to another, you're extracting energy, pumping out hydrogens, to this outer structure, to the inner structure, and what happens is you build up this tremendous gradient. And when you have the hydrogens going through a particular molecule, which I'll show you in a minute, it generates 90% of the ATP for the body use. Now, there are protein pigments called cytochromes. They're embedded in the mitochondria inner membrane. They transfer the electrons of the hydrogen atoms from one to another. It's kind of like the first one takes the energy and goes to another one, to another one. Think about it this way. If you had you and all your friends, and each one of you was on a step in a stairwell. So one person at the top hands a ball to the next person at the next step. That person turns and hands the ball to the next person down on the next step. And you keep going through this until basically a lot of the energy is released. And in the process, you're going to have this fantastic redox reaction, and you're going to have also a lot of hydrogen atoms or hydrogen ions pumped out to one side, and you are going to end up with a lot of capability to produce lots of ATP. By the way, when I talk about oxidation, keep in mind that that is the gain of oxygen or the loss of hydrogen or electrons. So what's reduction? The opposite. Loss of oxygen or gain of hydrogens or electrons. Now, this is where we get into the redox reactions. This sounds great, except do I have to know all the little itsy bitsy steps? No, not necessarily, but you do have to have some appreciation. There's two terms I want you to get familiarized with. Chemiosmosis. This is kinetic energy of passing of hydrogen ions to generate ATP. They have what is called the chemiosmotic gradient, and what you have is lots of hydrogen on one side, very little on the other. So what are the sluice gates that allow it to go through the membrane to the other side where the concentration's lower? That's where you have this unique enzyme called ATP synthase. And it's basically, as the hydrogens pass through it, it rotates. And as it's rotating, it's grabbing ADP, adenosine diphosphate, attaches another phosphate to it to make ATP. You see, the enzyme in the middle of it is an ion channel. And I'll show you a little bit about this. So here, we have the inner membrane space, we have the membrane itself, and we have all of these wonderful hydrogen ions that build up in this space. We have all of these cytochromes here embedded into this membrane. And then you have on this side here, the ATP synthase. Lots of hydrogens here, very few hydrogens here. So what happens is all these hydrogens start trying to come out following the concentration gradient. And as they do, they keep making ATPs, a lot of them. Now, what happens in the end with the hydrogen ions and then also this bit about we need oxygen? Well, if you remember your basics of aerobic respiration, cellular respiration, you have glucose plus oxygen gives you carbon dioxide and water. Bingo. So what I end up making at this end part here is hydrogen ions that meet with an oxygen ion, and you end up with water being made. The carbon dioxide was made in the other steps. Here's where you have the inner and outer membranes of the mitochondria, another good image. You have the proton gradient here. You have a buildup of lots of hydrogens here. There's not a lot of hydrogens down here. There are some, when they say it's chemiosmotic, you have still negative charge as the electrons eventually build up here. So the negative charges here and the positive charges on this side, they're going to attract one another. The concentration gradient is going to be there also. It's going to contribute this attractive force to draw the hydrogens through because there's lots of hydrogens here, very few hydrogens here. And when those hydrogens come down here, by the way, they get sequestered with oxygen to form water. So this process continues constantly. Here's the ATP synthase, and as the hydrogen ions run through it, it will basically take ADP, fuse a phosphate to it, and now you have lots of ATP. Now, in glucose metabolism, the glucose catabolism, I should say, yields 30 to 32 ATP. So you're breaking down glucose and getting a lot more energy out of it. NADH and FADH2 production will help yield some of the energy for more ATP production. Glucose is the energy fuel. There are other forms of nutrients that can be converted to glucose or enter other steps of the aerobic metabolism cycle, such as the Krebs cycle, etc., as I mentioned to you before, and they can be used to make ATP. So let's take a look at this in the big picture here. If you take glucose, you run through it, you get 2 molecules of pyruvate, they get stripped off a couple of carbons, and you go through the citric acid cycle, you get a little bit of ATP, you get a lot of these hydrogen carriers, which deliver their hydrogen to the electron transport chain. And the buildup of hydrogen will provide you with also further energy to make lots and lots of ATP. And if you look here and you add it all up, what do you get? 30 to 32. They used to say 36, by the way. But basically, it depends on the molecules and several other factors. But the bottom line is this: the electron transport chain is the final step. This is all part of inside of the mitochondria. This is all part of aerobic metabolism, and you get a lot more ATPs made as a result of this process, 30 to 32. Compare that to just glycolysis. You only get 2, not much. Now, nutrient metabolism follows several pathways. You have to keep in mind that cells need to build or store compounds. That's the anabolic part. Now, by the way, when I say store, that's where we get into what I was talking about before. Glucose is put into the liver, and it happens to be stored up as glycogen. Same thing in the skeletal muscles. Fatty acids can be used in adipocytes, fat cells. They will build up lipids in those fat cells. Then we have the catabolic. We extract energy from these compounds. Now, there's a couple of terms you do need to be aware of. First, gluconeogenesis, neo-new. This is the formation of glucose from non-carbohydrates, such as breaking down amino acids or breaking up fatty acid chains. Glycolysis is the breakdown of glucose to 2 or 3 carbon molecules. Okay. Glycolysis happens, and then you have also glycogenesis, which is the formation of glycogen from glucose. Glycogenolysis is the breakdown of glycogen to glucose. So in other words, when you talk about the glycogenolysis, what you're doing is, think about it this way: a big long chain of glucoses, and that's what glycogen is. And you chop off one at a time by enzymes so you can free up the glucose to be used for metabolic needs. And you can see all of this diagrammed here. If you're going to go in a direction to be anabolic, you take glucose and you make it into glycogen. That's glycogenesis. If you're going to take the amino acids, make them into proteins, okay, that's fine. But you can also break the proteins down to subsequent amino acids and take those amino acids, remove the amino group, and you've got yourself 3 carbon chains that you can then access for energy. Okay? Now, digestion involves a series of steps to make nutrients available to the body. And I basically just put down review the digestive process. So basically, let's just go through this really quickly and briefly. When we talk about digestion, we have to have mechanical grinding by teeth and tongue. We're going to basically make the initial breakdown a mechanical one, because the smaller the particle, the easier it is in the digestive system to break it down to the subsequent amino acids, monosaccharides, lipids, things like that. You're going to have some dissolving action by some of the saliva, particularly from salivary amylase, but not a lot. But it is the beginning of carbohydrate and lipid digestion, because you do have a little bit of salivary lipase. But again, we swallow things. We don't, sit there like a cow all day just chewing on one bite of food. We transport it down to our stomach, and there we have continued physical and chemical breakdown. Remember that the acidity also will denature proteins, and pepsin is the initial enzyme that will begin to break up some of these long chains of amino acids, which are really the proteins. We end up forming acidic chyme. This will then get pumped out of the stomach into the duodenum, and then we start the process of digestion further in the small intestine, the duodenum, then the jejunum, then the ileum. Okay, this is where also the pancreas will discharge certain enzymes to break down carbohydrates, nucleic acids, lipids, and proteins. We need to emulsify a lot of the lipids so that they're in smaller globs so that lipases can break down the lipids. That's where we have bile discharged, which acts as a emulsifying agent. And so we have smaller globs of lipids that then break down further lipase down to things like monoglycerides and fatty acids. Now, what happens here in the small intestine? You have nutrient absorption, and it's transported in the blood, the amino acids and the monosaccharides. The monoglycerides and the fatty acids are usually too big and can't get transported by the blood anyways, at least not initially. So they get picked up by the lacteals. These are small, basically parts of the lymphatic system, lymphatic capillaries, if you want to call it, and they get transported eventually through the lymphatic system and eventually deposited into the blood. Okay. Now, all of this process is occurring, and you also made or discharged a certain amount of water. So what happens is we eventually have what would be a pasty material, somewhat fecal-like, that gets discharged from small intestine to the large intestine. There, the water is reabsorbed. There, bacteria will help generate further organic nutrients and vitamins. Those vitamins will be absorbed. And eventually what happens is that you form the fecal matter, which is excreted. The fecal matter will contain undigested polysaccharide fibers, things like cellulose, stuff like that. It'll also contain about 30% of it will be bacteria cells. And we may also have some other materials that were not well digested that are excreted. Okay. Now, the absorbed nutrients that are in the blood will be passed and transported to the hepatic portal vein. The liver will absorb the needed nutrients. The rest of them will be passed on to the rest of the systemic circulation. The cells in the peripheral tissues will absorb the nutrients. Everything looks good. A couple of pointers I do bring up to you here in this slide. Remember that you're going to have really the times in which you're going to have digestion, and then times that you have to have specific breakdown of products so that you can eventually get ATP production. How do you get it under resting conditions? Well, we're going to go into that a little bit more in a few minutes. The carbohydrate digestive enzymes you have are amylase, which is both salivary and pancreatic alpha amylase. Disaccharides are usually what you end up with after amylase has done its job initially. And there is the brush border enzymes. These are ones that are located in the microvilli. They are referred to as maltase, sucrase, lactase. Maltase, if you remember, maltose is a disaccharide. That's basically two glucoses together. Sucrase is fructose and glucose, and lactate is galactose and glucose. They have to be broken down to individual monosaccharides to be absorbed. Now, if the carbohydrates are not digested and absorbed, we face a situation called flatus, which is an intestinal gas. Yes, I know it's embarrassing, and some people are, make jokes about it, like we're all little kids and so on and so forth. But see, flatus can also be an indication of some improper digestion too. Okay, for example, you have inulin, which is a heterogeneous collection of fructose polymers. They're found in root fibers, for example, the Jerusalem artichoke, bananas, wheat. These can cause flatus. Here's the key point to help you. Anything that doesn't get broken down in the small intestine is fair game in one sense or another, for the bacteria in the large intestine. Now, if someone has a nutritional deficiency or has a enzyme deficiency, for example, there are those that have lactose intolerance. What it means is that they're not making lactase anymore. So the lactose, this disaccharide, goes from the small intestine, does not get digested, goes into the large intestine and becomes fuel for the bacteria. That causes gas, cramping, and some cases, in extreme, it can cause diarrhea and such like that. You have others who have improper digestion of fats. So what happens is they have what's called stearorrhea. And what that means is that the lipids are not being broken down. The lipids pass into the large intestine and eventually pass out. And literally the poop will float because of the amount of lipids that are present. Okay? Anyways, as you can see here, this is where we talk about the breakdown of carbohydrates as it goes through. Now remember, carbohydrates, when we talk about monosaccharides, the liver is going to store some of them as glycogen, and this will be released out over the period after you've had your meal. In other words, for the other 5 to 12 hours that you haven't had a meal, the liver will break down the glycogen by certain hormonal signals and other signals to reverse this and keep blood glucose levels elevated to the certain amount that's necessary. Glycogen is also going to be built up in skeletal muscle. Let me get you to think about this. You've heard we've had the Boston Marathon in the past, and they have, usually on the weekend, just before the event, they used to have certain pasta makers, particularly Prince Spaghetti, and they would do carbohydrate. The runners would do this. They would do carbohydrate loading. What it is that they're trying to, in the few days before the race, they reduce their running, they increase their carbohydrate intake, and this produces larger amounts of stored carbohydrates so that when the race occurs, they will then be able to adequately have energy. Because at a certain point in that marathon, they've run out of their glucose reserves, and so they start to then metabolize lipids and proteins. Yep, they will actually start to break down some muscle proteins if necessary. But we'll continue with this later. Now what we're going to do is focus on lipids and the lipid metabolism. Okay? So first we have to talk about this from the perspective of lipids that reach the bloodstream. How can you get lipids, which really are not polar molecules? They don't mix well with water. How can you get them in there? Well, you start forming these chylomicrons. Then you have cholesterol is then extracted and released as lipoproteins. This whole process starts off with, of course, lingual and pancreatic, lingual meaning in the mouth, lipase. Now, all lipases split triglycerides to monoglycerides and fatty acids. Bile salts, as I mentioned before, are a emulsification process. And these are amphipathic molecules, meaning that part of the side of the molecule is polar, part is nonpolar. They start forming these little micelles, which are small lipid and bile salt complexes. Okay? In other words, instead of a big blob of lipid, you've got these smaller ones, just like a detergent when you're using to clean greasy plates in, when you clean your dishes at the end of the day. Now, when we start breaking down these lipids, we transport them by the lacteals and the lymphatic system, which I mentioned. We have what are called eventually, we form these chylomicrons. These are protein-coated complexes with insoluble proteins. So in other words, the proteins have the capability to grab onto the lipids, and the proteins will face the watery exterior of the environment, which allows them to be transported in the blood. Now, lipoprotein lipase, this is found in capillaries and breaks down the chylomicrons to fatty acids and monoglycerides for tissue uses. Okay. The liver also will make what we call LDLs and VLDLs. Now, HDLs, let me get you introduced to this. are high-density lipoproteins, meaning that there's mostly protein present. Think of it this way, it's like a glob. Okay, you're going to have some of the proteins there. You're going to have some of the lipids there. Some of the lipids may be fatty acids, some of them may be cholesterol, some of them may be this or that, monoglycerides, etc. Now, how do you tell the difference between HDL, LDL, VLDL? HDL, you're going to just basically say it's more high-density lipoproteins, okay? And the higher the number of your HDLs is really a good indicator as compared to your LDLs and VLDLs. Now, LDLs are low-density lipoproteins. The liver will remove the triglycerides and it adds cholesterol. VLDLs are really the difficult ones because they're very low-density lipoproteins. They have a certain amount of cholesterol, some triglycerides, and phospholipids. So when we all started trying to figure out, and this started in the 70s, how do we look at the risk of heart attack? And then how do we look at the risk of atherosclerosis? Because what happens is you get the delivery into some of the blood vessel walls of some of these lipids. And in the beginning, it was sort of focused totally on LDLs, then focus LDLs and VLDLs. And now what they try to do is look at the LDL and HDL ratio, because the idea being is if you have a high HDL number, the cholesterol number doesn't seem to have as much of an impact because you have the capability to pull some of these out because of the abundance of HDLs. And there are certain activities, certain diets that do improve the HDL levels. Now, the other problem is on the other side, okay? High cholesterol numbers, high LDL and HDL ratio, meaning there's a lot more LDLs than HDLs, means you have too much cholesterol in circulation, too much cholesterol going to the tissues instead of recycling it or excreting it. Yes, you do excrete some cholesterol. And that's where, if you remember a while back, they used to have all these Cheerio commercials, having Cheerios in the morning because it's oats. And it's a soluble fiber, and what it does is it binds to cholesterol. Yes, it will do this, but it's not like if you ate, an oat field that you would suddenly have your cholesterol numbers drop. You have to have this on a consistent basis, A. B, you also have to modulate your diet so that you're not just having lots and lots of high cholesterol. And a lot of people would have differing views on that. The one thing I would throw out to you is always be in consultation with your physician. Sounds like a legal statement, but you really have to keep that in mind because your physician knows your personal health better than anybody else, okay? So let's move on from here. And you can see this entire process, I would ask you to review this, of the process of lipids, the lipid metabolism, the lipid transport, okay? It's extremely important to get down what choliomicrons are, the VLDLs, the LDLs, etc., because you have to keep in mind something. There was two things that are very important here I want to bring up. One is that lipids are necessary as part of your diet. Lipids are necessary because they are the building blocks to help make cell membranes. And unfortunately, I've mentioned this before, that you have some individuals who had basically went on these ultra low lipid diets and ended up terribly ill. And increasingly, they had gallstones because the bile would just sit there and concentrate until it crystallized and formed stones. Okay. The other point is this: you have individuals who have hypercholesteremia. And that is because the cholesterol receptors in the liver do not pull the cholesterol out. Now, you probably have heard where it's important to try to keep your cholesterol numbers, depending on the age, below 200, 230 as you get into your 50s and 60s. These people had 470, 480, 500 numbers. These were people who had a gene defect, and as a result, could not pull their cholesterol out, and a lot of times they ended up dying very early in life. And 30s, 40s. lot of atherosclerosis. Really sad. But anyways, let's move on. Now, let's talk about how we metabolize lipids, because we were going through all of the carbohydrate stuff before, but let's talk a couple of points that are very essential. Fatty acids can be broken down to provide energy, yeah, and they're converted to other lipids. Okay, so what is lipolysis? Lysis is to break. Okay, so these are lipids broken down into fatty acids, monoglycerides, or glycerol for energy production. Now, how is this process occurring? This is where we get into what's called beta oxidation. This is a catabolic process of the fatty acids. Think of fatty acids as long chains, and every once in what you have are enzymes that just come in and go glunk, cut off 2 carbon compounds. Glunk, 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 glunk. And when they do this, the 2 carbons get incorporated with a coenzyme, and you form acetyl-CoA. And if you remember, acetyl-CoA works very nicely going right into the Krebs cycle and thereby producing the energy. And that's how you get energy from lipids. Now, lipogenesis is the synthesis of lipids. Here's one important point. There are a lot of lipids that we can make ourselves, but there are some that we cannot and are absolutely essential in our diet. These are referred to as essential fatty acids. Fatty acids not synthesized by the body and must be incorporated into our diet for proper health maintenance. These are usually fatty acids made by plants, and they can be used to be synthesized into, for example, prostaglandins. These include linolenic acid and linoleic acid. You have omega-3s and omega-6s. Sorry about the minor typo there, but it's omega. Now, the omega-3s and the omega-6s, everybody's gotten into this wild idea about, I consume a lot of that, I should not have a heart attack. No, it doesn't work like that. But what you have to keep in mind is these are the building blocks necessary for a variety of cell signaling compounds. We generally call them prostaglandins. And they're necessary. If you take a look here, this diagram takes, remember the fatty acid chains here, and you can see where the dark areas are, that's carbons. So basically, if you're going to lop off a 2 carbon molecule, you lop it off here, lop it off here. The 2 carbons join with coenzyme A, and what you literally have is acetyl-CoA, which is put into the citric acid cycle, and their products are also eventually sent into the electron transport chain, and you get lots of ATP out of it. On top of that, you can take glucose, and at a certain point of the glycolysis, take out and pull the 3 carbon molecule out and basically make it into glycerol, which with fatty acids makes triglycerides. The fatty acids can be used to synthesize cholesterol, prostaglandins, phospholipids, glycolipids. Now, there are a few prostaglandins that need that omega-6 or omega-3. Cholesterol is also the building blocks to make steroids, okay? Keep all of these things in mind. Now, what about proteins? You hear people talking about this, okay? You have to understand that an amino acid that's not needed for protein synthesis is not going to just be stored. It will be either broken down or converted to a different amino acid. Those are the amino acids that are considered non-essential amino acids because we can flip them from one type to another. Digestive enzymes break down proteins into amino acids. The first digestion step is, of course, in the stomach with pepsin, and then we have the duodenal epithelium, which has the entropeptidases. You also have, made by the pancreas, trypsin. Trypsin will be converted from trypsinogen, which is the inactive enzyme. And then active trypsin will activate all the other enzymes, the chymotrypsin, the carboxypeptidase, the elastase, and the dipeptidase. All of these will get activated. Once they're all activated, they're all starting to break down the proteins in the space of the duodenum and furthermore downstream. Chymotrypsin, carboxypeptidase, elastin, just like trypsin, are made in the pancreas. The dipeptidase is a brush border enzyme. So we've made these nice little amino acids, but we can't make all of them. There are 10 essential amino acids. They can't be made in the body and they have to be supplied in the diet. We have to also understand a couple of other steps that are absolutely critical when we talk about amino acids. Amination is attaching an ammonium ion to form an amino acid. Transamination is moving an amino group from one molecule to another to form an amino acid. Now, wait a minute. Didn't you say there were essential ones? Yes, but I'm talking about the non-essentials. Okay? We'll get back to the essentials in a second. When we talk about amino acid catabolism, the breakdown of amino acids to obtain energy, what you have to do is you have to remove that amino group. So we have what's called deamination. It's the removal of the amino group from the amino acid, and it will form an ammonium ion. Now, the bulk of this occurs in the liver. One problem, ammonium ions, or ammonia period, is toxic to cells. So what does the body do? Ammonium ions are joined together with carbon dioxide to form urea. Now, in human beings, mammals, we have a water-soluble nitrogen waste called urea. We're going to touch a little bit about this toward the end, but if you remember, I mentioned this in class once before. Bird comes by, poops on your car, you got that white crystalline stuff, and you're all like swearing at the bird, especially with a brand new waxed car. You'll notice that when you pour water onto the poop, the white stuff doesn't go away. That's uric acid. It's not water soluble for the most part, okay? This is how birds, reptiles, amphibians, etc., they get rid of their nitrogen, but it's in a form of what is called uric acid. We make a little bit of it, and we can have too much of it and it leads to gout. But uric acid is not water soluble. Our nitrogen waste is water soluble, urea. And the urea cycle, the reaction sequence responsible for the production of urea, occurs, as I said, in the liver. And you can see all of this occurring here. All right. Now, amination, you take something, let's say, like alpha ketoglutarate, add the ammonium, boom, you got glutamic acid. Let's say you want to take glutamic acid and convert it into tyrosine, you can do that. You can move the nitrogen over. That's using transamination. But let's say you want the glutamic acid just floating around, it's not doing anything. You can turn it into fuel by deamination. And what you end up forming is an organic acid product that you can break down further. And you have the ammonium ion. But as I said, the ammonium ion is toxic. So you immediately start sequestering it, forming chemical bonds with carbon dioxide, and you form urea. That's the urea cycle. Don't sweat it. Know it's a cycle. I'm not going to have you understand all the enzymes and all the steps. Again, that's for a biochemist. No problem. Now, there are two general patterns of metabolic activity, the absorptive and the post-absorptive. This is where we get into what I was mentioning earlier about why we end up storing a lot of glucose in the liver in the form of glycogen. We have the absorptive state. That's the time following a meal when the nutrient absorption is underway. You had a wonderful meal and you have to now, you're breaking it down, the proteins, the carbohydrates, the lipids, and it's going to go getting transported in either the blood or the lymphatic system, particularly if you're talking about lipids. Insulin is the primary hormone during this state. Now, we've talked a little bit about that before, but let's talk about now the post-absorptive state. This is a time when nutrient absorption is not underway and the body relies on the internal energy reserves to meet energy demands. Another way of saying it is we're going to use our fuel that we've stored away. Now, there are different hormones that play a role in this. Glucagon, epinephrine, glucocorticoids, growth hormone. Do I have to know this? Yes. Okay. This is where we start looking at the various fuels and what is necessary, what's needed, where is it stored, and where does it go. Glucose. This is going to be released by the liver into the bloodstream. You're going to break down glycogen into the individual glucose molecules and get them into the bloodstream. Remember, What's the organ that absolutely needs glucose? Doesn't take anything else? The brain, the nerves, the neurons. Fatty acids are released into the bloodstream by adipocytes. Ketone bodies produced by the liver cells but are used by peripheral tissues. Amino acids are released by the liver and skeletal muscles from proteins that are broken down. And you can see all of this as it occurs. Okay, that looks nice. When we need these fuels, we can break them down. And what happens here is insulin will stimulate glucose uptake, glycogenesis, amino acid uptake, protein synthesis, triglyceride synthesis. Insulin will also act on androgens, estrogens, and growth hormone, also will stimulate the protein synthesis. Glycolysis, aerobic metabolism, provides the ATP necessary, as well as the synthesis of lipids and proteins. That's in the absorptive state. Now we go into here, post-absorptive. Glucocorticoids are going to stimulate the mobilization of lipid and protein reserves. These effects are enhanced by growth hormone. Glucagon will stimulate glycogenolysis and gluconeogenesis, making new glucose, breaking down glycogen. Primarily, this occurs in the liver, and what you're going to do is release a lot more glucose by the liver for the rest of the body. You're going to shift away from glucose metabolism by other tissues so that you have a stabilized blood glucose level. Epinephrine is important in stimulating glycogenolysis in skeletal muscle, cardiac muscle, and lipolysis in adipocytes. Now we move from here to start talking about a couple of other compounds, and this is where it gets sometimes a little bit emotional because there is such fluff in pseudoscience when you talk vitamins and minerals. What I mean by pseudoscience is somebody suddenly says, hey, I'm going to tell you about a new vitamin that's supposed to be going on there, or hey, I'm going to tell you about these minerals that the government doesn't want you to know, or hey, and they tell you all of this stuff, and in the end, they're trying to sell you something, okay? Or it's a fad, okay? Keep in mind, what are vitamins and minerals? Vitamin is an organic compound required in very small amounts, but used in specific metabolic pathways, period. Minerals, these are inorganic nutrients, okay? They serve as electrolytes in body fluids. They also may be a cofactor in some enzymes. Give you an example. Okay, electrolytes, well, that would be like calcium, sodium, potassium, chloride. Fine. What about iron? Well, iron is used in hemoglobin, and it may be involved in some functions of certain enzymes. What about iodide? Well, iodide's incorporated into thyroid hormone. That's important. Okay, but then you have a variety of other minerals that are also in very, very small amounts, but are essential for the function of some enzymes, period. Now, there's two major classes of vitamins, fat soluble, and this is where the vitamin is actually soluble in fats and they're stored in adipose tissue, and that's going to be vitamins A, D, E, and K. That's it. Water-soluble vitamins. These are vitamins that are soluble in water and tend to be excreted in urine. So it's a lot harder with a water-soluble vitamin to get an overdose because it'll be passed out. And those are the B vitamins and the C vitamins. So all the B vitamins, B1, B2, et cetera, as well as vitamin C. Now there's two other concepts you got to keep in mind here. Hypovitaminosis, which is a vitamin deficiency disease. You don't have enough vitamin A, you don't have enough vitamin D, you don't have enough vitamin C. Hey, don't have enough vitamin C, you have scurvy. Okay. Hypervitaminosis, which some cases occurs a lot more in our society because people will buy a whole bottle and say, hey, a little's good, a lot will be better. Nope. Is vitamin overconsumption that exceeds the body's ability to excrete the excess and leads to bodily disease. You can overdose and cause a lot of damage to the body by having too much vitamin A or vitamin D, okay? And this happens a lot. Oh, one other thing, you want to keep in mind that Most of the B vitamins, the water-soluble vitamins, are absorbed on their own, but there's one, vitamin B12, that needs a unique compound. That's that glycoprotein that I mentioned to you about before, the intrinsic factor that's made in the stomach. It's necessary for vitamin B12 absorption. And as I mentioned to you before also, you have sometimes elderly individuals that they don't make as much intrinsic factor. So as a result, they tend to have the possibility of vitamin B12 deficiency, which is important in the synthesis of red blood cells, as well as making of DNA. So they tend to have to then give them a injection of vitamin B12. Okay. Here is the example of vitamins A, D, E, and K. And look, the artist just took these little nice pictures here, saying which do you want your vitamins in? Fruits and vegetables and all these other wonderful goodies, or in a pill. Okay. These do not necessarily tell you that all of these wonderful fruits and vegetables are going to have lots and lots of these vitamins. You would find a lot more vitamin A in things like fish products, green and leafy vegetables. Usually it's because of the bright vegetables make carotene, which is a precursor to making vitamin A. Okay. And you would find other vitamins in other types of food. So in other words, diversified diet enhances the health of the individual by having a greater chance of meeting all the nutritional needs of both vitamins and minerals. Here is the water-soluble ones. So everything that's B, from B7, biotin, B12, of course, B12 needs, of course, also that intrinsic factor. Folic acid, pyroxidine, pantothenic acid, niacin, riboflavin, thiamine. If you notice, here are effects of the deficiencies of it, and then, of course, vitamin C. Okay? Now, we're going to move into a basic concept of food, nutrition, intake, etc. And this is very interesting because, when proper nutrition depends on having a balanced diet. But balanced diet is not everything. You've got to have physical activity. In other words, you take in a certain amount of calories, you've got to balance it off with the caloric consumption that occurs. If you have too much activity and not enough caloric intake, then you're going to be having calorie deficiency. If you have too much calories and not enough activity, then you're going to start building up weight. Now, know the limits of fats, sugars, and salts. That's something that's been repeated constantly because we know that saturated fats, trans fats, and sodium are not necessarily good. But at least we understand this a lot better than when they first started to say this. And of course, trans fats have been pretty much eliminated from our diet because of the fact that trans fats, which are partially hydrogenated, tend to increase the risk of cardiovascular disease. Okay? Let's talk about this a little bit more because really a proper nutrition depends on eating a balanced diet. When I say a balanced diet, someone would jokingly say, well, look, I'll have my steak on a high wire. Oh, that's a bad joke. Sorry, guys. Basically, you're containing all the ingredients necessary for homeostasis. Malnutrition is the unhealthy state resulting from inadequate or excessive absorption of one or more nutrients. Here's where we get the little tricky part. Now, if you had some physics or you had some chemistry, you may have understood this. When we talk about a calorie of energy, this is a small amount of energy. It's the energy required to raise 1 gram of water 1 degree Celsius. So what's that difference between that and 1 calorie of food energy? What happened was people mixed that up. Because when we talk about 1 calorie of food energy, it's basically 1,000 energy calories. You're kidding me. So if I go out and I have a Big Mac and I have 325 calories, I'm really consuming 325,000 calories of energy? Yeah. But what happened is to help the average consumer, they don't call it the actual true energy calculation. They call it food energy calorie. and that's basically the equivalent of 1,000 energy calories. In the next couple slides, I'm going to show you the history of food dietary plans. Okay. Before I go into that, I want to go over one other little point. This is called complete proteins. Complete proteins usually are going to be animal-based. They're foods that contain all essential amino acids. Incomplete proteins are foods that lack one or more essential amino acid and have to be mixed with other foods to maintain diet containing all 10 essential amino acids. Case in point, beans and corn. Why do people have beans and corn together? Because basically one amino acid is deficient in the beans, one is deficient, but a different one in the corn. You mix them together. I think corn has a very low amount of lysine as one of the amino acids, but that's compensated when you mix them together. Whereas if I had a steak, I'd have all the amino acids. What happened was people started understanding the difference between complete and incomplete. Incomplete, and for those that like to be vegetarian or vegan or whatever, that's your choice. The thing is, you got to make sure that you're going to properly have the sufficient amounts of minerals and vitamins, as well as having a proper mixture of incomplete protein foods so that you get fulfilled proper dietary intake of all the necessary amino acids. This is what we see today. We see today myplate.gov. This was implemented about June of 2011 forward. Usually what sets this up is the USDA. Let me give you a little bit of background here. Public health was influenced after the Great Depression. You see, when World War II started getting when they started revving up all the troops and doing the draft and everything, one of the problems that they had was for about a decade of the Great Depression, people were starving. They had malnourishment, they had all sorts of deficiencies, and as a result, you had lots and lots of young people that now showed up with poor health, couldn't be incorporated into any military service, couldn't barely function as well. Now, so the USDA and various other government sectors started saying, look, we've got to help teach the people how to properly eat. If you go back 500 years ago, beans and hardtack was really just very tough bread, was basically the diet for everybody. Nobody thought about breakfast in the sense of, oh, wow, I want my Cheerios and milk. As a matter of fact, just to give you a little balance there, having a cereal with milk, for those that can consume milk, is very important, as I've mentioned in ANP1 before, because you have the phosphorus that comes from the grains and the calcium that comes from the milk, and you bring them together, and that permits for proper bone growth. But nobody knew that in 1900, let alone 1500s. Now, this had changed, but you still see the major groups here: grains, protein, dairy, fruits, vegetables. Now, go back before then, basically from 2005 to 2011, what you see is a my pyramid that has a certain amount of the different food groups, but it also incorporates a certain amount of activity. This happened to be a change from the food pyramid, which was implemented from 1992 to 2005. If you notice there, you have the proteins, you have the various fruits, vegetables, etc. Now, they would tell you the servings per day. But you know what was a problematic little issue here? Well, simple. At the top of the pyramid, we were told to avoid lots of sugar, refined carbohydrates, so flour, pasta, grains, potatoes, really a lot of stuff like sugar, salt, and and fats, saturated fats. Now, this is a modified version of the food pyramid, but there are very basic ones there. And the problem was that if you were sitting there saying, well, hey, I had my broccoli today, but I dipped it in melted cheese. Well, there you start seeing you're having a lot more fats brought into the diet. In other words, it can sneak in. Well, hey, I'm going to have my whole grain, but I'm going to have it dipped in, like um, Philly cheese, or I'm going to have it in cream cheese, or I'm going to have it with this or that. And so it got to be very difficult at times to coordinate that. Plus, although people were in some cases following this food pyramid, their activity level was still low. Yes, they were couch potatoes. Yes, they had sedentary jobs in front of computer screens all day and night. So they started picking up weight. Now, this is 92. Let's go back before then. This is what you face for your parents and probably your grandparents for quite some time. It was called the Basic Four Food Groups. This went from 1956 until 1992. Meats, dairy, fruit and vegetable, grains. Not telling you the amount, and certainly not bringing in the metabolic activity that would occur. Let's go before then. This is what they saw, and they called it sort of the 7 basic groups. And it was from 1943 to 1956. Now, as I said to you, part of this was a, if you want to say, reaction or response to a lot of the people coming out of the Depression were in poor health because of poor diets. Nobody sat there and said, hey, you should have fruits and vegetables as well as your bread and your butter and maybe a little bit of meat. They didn't care. They couldn't afford things like fruits and vegetables in some cases. Or in some cases, they'd have fruits and vegetables, but they would also have a lot of butter and milk. And you can imagine all of this and how it would make for someone to be growing up unhealthy. Think about it this way. Somebody born in 1925, and if you look at that 1945, they would have been 20 years old. If World War II was going on, let's say 1943, they would have been 18, they would have been draftable. And in some cases, if they went through a good part of the Depression, the time in which somebody was growing and developing, they were going to be malnourished through that point. And it might mean lower body mass, weaker bones, less intellectual capability. Doesn't mean everybody suffered, but there was quite a few individuals that really had a difficult time in those periods. Okay, let's move forward here. And I brought up the 5 basic groups, the effects on health. I would just ask you to review that basically. You have on one side complete proteins versus other side incomplete proteins. Now, of course, they don't mention in this photo here. It's more fruits and vegetables. They don't have included into it the grains, etc. It is the mixing of the grains and other types of seed products that may help to deal with making complementary protein foods and meeting the needs of all of the 10 essential amino acids. That's the best way to put it for you. Okay. Okay. Next, we're going to be dealing with is metabolic disorders. They may result from nutritional or biochemical problems. Some of these disorders are related to diet and digestion. Some of them have a psychological component. First one would be anorexia nervosa. This is a psychological lack of appetite, a loss of weight, and this can be very, very fatal. A lot of times you see this in Western young females, but you have seen it in other parts of the world too. The young ladies think that they're still too heavy. They will do excessive amounts of exercise and very low amounts of eating. And so what happens is literally they're losing weight and basically they're becoming weaker and sicker. Okay. The eventual situations occur when they start having difficulty controlling their body temperature, when they have lost muscle mass, when they become amenorrheic, meaning that the menses have stopped, etc. Now, it can be turned around, but it's sometimes a battle for these individuals. Bulimia is the over excessive eating, that's the binge, And then you have the following a purging, which is usually induced by vomiting, and it may include also use of laxatives and diuretics. This is a big problem because this can be fatal, because it will lead to electrolyte imbalances due to the vomiting, as well as esophageal ulcers and tooth erosion because of the acids from the vomitus that are destroying the tissue. The next area is obesity. And you have two major types. Regulatory obesity, usually it's a failure to regulate food intake with caloric needs. Then you have metabolic obesity. This is a condition that underlies bodily malfunction of the metabolism. This form is much more rarer than you'd see. Now, if you see here, here's an example of anorexia. Here's an example of obesity. And here we have a situation where we start building up cholesterol deposits into the blood vessels. And that is due to excessive cholesterol deposition from the low-density lipoproteins. So that brings us up to the elevated cholesterol levels. Elevated cholesterol can lead to atherosclerosis and heart disease. There was a series of commercials done a while back where they would take some silliness. It was very interesting how they would do the photographic segueing. You know, they basically were saying to individuals, look, it's not just what you eat, but it's your relatives. Yes, to a point, your metabolic capabilities to use calories can come in part from genetics. So you'd have this, you know, it's not just what you ate, but it's your Aunt Selma, it's your, you know, grandfather, as well as that. One of the things that I found that was interesting is one of the things that was said by some physiologists and some psychologists. They said, if you really want to get an idea of how you're going to look, your body, your metabolism, etc. Well, one, keep in mind that basically your metabolic rate after age 30 decreases each decade by 2 to 5%, depending. But the other thing is, they said, don't look at your parents at whether or not you're going to be thin or more on the portly side. Look at your grandparents. And that's very true. I've seen that myself. Anyways, nutritional and metabolic disorders. Here's an interesting one. Phenylketonuria. Now, any of you out there right now that are drinking something diet Coke, Diet Pepsi, look on the side of the can and it will say, phenylketonuria should not consume this. Why? Phenylketonuria, PKU, is an enzyme gene defect. And basically, the individual cannot metabolize phenylalanine. They can't convert it from phenylalanine to tyrosine, because tyrosine would then be, of course, the precursor to make a certain types of catecholamines like epinephrine, norepinephrine, dopamine. Instead, if the enzyme's not present, you end up with another enzyme that does a side reaction and produces a toxic product. Now, here's the problem. In the 1980s, we started coming up with these artificial sweeteners. One of them was called aspartame, otherwise known as NutraSweet. That's the commercial name. And really what it is it's a combination of aspartic acid, one amino acid, and phenylalanine. And what happened was this. Phenylketonuria earlier was identified. And what happens is we all go through this when we were born. They take a little blood from the heel and they do a little test immediately at birth. And if you have PKU, they nowadays would just tell you, okay, that's what you got to do. You got to avoid these foods, and these foods, and you'll be a healthy, happy person. Beforehand, individuals that would have this, that were PKU, that didn't have the testing, didn't have the modification of the diets, tended to end up being mentally handicapped because of the toxicity. Now, there were, in the 1980s, individuals, they're drinking, chugging down Diet Coke and going, Doc, I don't know what's wrong, but I don't feel well. And they look and they go, well, this guy tested when he was born, PKU, scratch in the head. What's going on? And they look at the Diet Coke and they go, well, let me see that. And Diet Coke says it. Don't consume this if you are a phenylketonuria. As soon as the patients would stop consuming this, they felt better. Okay. Next, protein deficiency diseases. This is usually where the protein synthesis declines throughout the body. Here's the thing, if you're malnourished, if you're starving, you're going to start seeing protein synthesis stop and being reversed. You're going to break down proteins to use the amino acids for fuel. But if you don't make these proteins, you are going to start seeing less and less proteins in the blood. Less and less proteins means the osmolarity of the plasma fails, and you're going to have edema, meaning more fluid will get into the tissues. That's going to cause bloating. Now, it's interesting with kwashiorkor, this is where the protein intake is inadequate. They may be getting a little bit of carbohydrates, roots, grains, something like that, but this is very common to see in very starving children. You see a bloated belly, thin body frame, as you see in the photo next. Now, by the way, the bloated belly is not because of fat. The bloated belly is because that's all edema. It's all fluid. And usually when they take these children and start to treat them, they have to give them sort of a peptone, in other words, protein fragment meal, not full protein. You can't give them a steak, they throw it up. Literally. They have to be given sort of a gruel that would be easier to digest, because remember, if they're not making proteins, they probably don't have a lot of digestive enzymes functioning. And what would happen is they give them these to build them up. Now, the other thing is that if they're not making proteins, one other thing, remember the immune system? What's the key defense? Antibodies. No amino acids, no protein, no antibodies. The immune system is compromised. They are usually going to be much more susceptible to disease. Okay? Now, the other type where they're just all stick, in other words, all thin frame, no bloated belly, everything, that's called marasimus, and that's just starvation, and the person is on the way to dying unless they can get the necessary treatments. Another area, ketone bodies. What ketone bodies are small organic acids resulting from lipid and amino acid breakdown. They can build up in the blood. Examples would be beta-butyric acid, acetone, things like that, okay? This leads to what's called ketoacidosis, okay? The buildup of ketone bodies in the blood causes a pH shift, and the pH shift is toward acidity. Now, you can tell this if someone is diabetes type 1. They're going to have a fruity smell to their breath. Sorry about the typo. And then you have ketosis, which is the acidification of the blood. So basically, the blood can go down as low as 7.05. Now remember, normal pH of blood is 7.35 to 7.45. So you're really going low on the acidity, and that's going to interfere with the function of proteins, etc., and the normal function of blood in and of itself. Okay? Now, uric acid. We've talked about uric acid before. It is very, very low in water solubility. It is a nitrogen waste. Usually it's due to the breakdown of adenine and guanine, the purines, and they're deaminated and excreted. But what happens is uric acid can build up. And sometimes what causes this can be just diet, can be having high amounts of purine-rich foods, etc. And then you have what's called gout. And this is the buildup and the crystal formation. And these are crystalline deposits of uric acid, and they build up in the tissues. Now, it's not just in like, a muscle of your arm or something. It's usually in the joints. And that's because, in part, they will stay in the joints and they won't always redissolve. It takes certain drugs to help them resolubilize and get them out of the body. This is where you get also not only gout, but what's called gouty arthritis. It is gout with inflammation in the metatarsal phalangeal joint of the hallux. That's the big toe. And you see that right here. Here is the Diet Coke. Phenylketouretics cannot consume. Here is an example of kwashiorkor. Notice the large belly, which is all edematous. And you have yet the frame. All of the muscle has been basically broken down. And this child is starving. Okay, Last area we're going to be talking about is energetics, and this is the study of energy changes and thermoregulation involving the heat balance. Now, I want you to get familiarized with the basic term, which is basal metabolic rate, BMR. This is the minimum resting energy expenditure of an awake, alert person. Now, think about this. You're sitting there right now just listening to this. You are still using energy. Your heart's beating, you're having respiration, so that means muscles are moving. You are processing information in your brains. That means that the brain's using glucose. You are also having your kidneys filter the blood, et cetera, et cetera. Even when you're asleep, these actions are all going on. So you're using energy, even though you're not doing a lot of physical activity, et cetera. Now, the other principle we're going to be dealing here with is going to be thermoregulation. That's the homeostatic control of the body temperature. Now, here you have an individual who is being monitored for their energy expenditures, etc. And they're working on an exercise bicycle. And so this researcher here is recording heart rate, metabolism, carbon dioxide production, oxygen retention, as well as monitoring all other basic physiological functions while these activities are going on. So calories per hour. When you're resting, maybe 75 calories per hour. Slow walking, you're getting up close to 200. And as you increase the physiological activity, the aerobic activity, you're going to continue to burn a lot more calories per hour. Now, we've been talking about using calories. Let's talk about the control of appetite. This is a complex system, and it involves both short-term and long-term mechanisms. Now, what you find is one of the primary areas is the hypothalamus. There are hypothalamic nuclei for feeding center, which relates to hunger. Ooh, I've got to eat. And satiety center, which basically says, ooh, I'm full, I'm sated. So this is where we have this center that says, related to food satiety. As a matter of fact, in some earlier studies in the 1980s, I believe 70s, they would take rats. They would basically ablate or destroy that particular satiety center, and you'd have this whopping rat, like a couple of pounds on a scale, weighing down the scale, because the rat had no feedback, no neural control on when satiety would occur. So it would just eat and eat and eat and eat. Now, why do we study all this? Well, diet, health, nutritional demands, etc. The dietary industry has been going bananas. The pharmaceutical industry has been going bananas trying to find the magic bullet that they can give to somebody, an inhibitor, and so they can lose weight. So let's look at the neurotransmitters involved in this appetite regulation. One of them is called neuropeptide Y. It is a hypothalamic neurotransmitter. It stimulates the feeding center and increases appetite. Now remember, appetite's not just something where you say, ooh, wow, I'm hungry. Remember, we have a lot of other social cues that influence us and how we eat. Come on, let's face it. You want to go on a date, you ask them to go out for dinner. You celebrate after a team wins, and what do? You don't sit there and starve. You go out and you have, pizza or an ice cream or whatever, right? So we use feeding as part of our socialization, as part of our responses. Now, I'm kind of sounding like I'm getting into sociology and psychology here, but there's a point, because we feed sometimes when we're not hungry, as long as we're not sated and it's filled. Okay? Okay. Now, ghrelin, which I've introduced to you earlier, is secreted by the gastric mucosa, and it stimulates appetite, whereas leptin is secreted by adipose cells, and it binds to CNS neurons and provides the sense of fullness and appetite suppression. And if you remember, I talked about this earlier in the course, where leptin, they found out when they first isolated it in the mid-1990s, they would sit there and say, hey, this is fantastic, look at what we've got. And what they were doing was studying morbidly obese individuals. The morbidly obese individuals were missing the gene for making leptin, so they had no real sense of food or appetite suppression. But then they had another subgroup of that they were studying that had leptin. They had very high leptin levels, and they were scratching their head going, what's going on? Turns out they had a genetic defect in the leptin receptor. So the defect in the receptor would be similar to, let's say, a radio antenna or radio receiver. The leptin would be the radio transmission. You get lots and lots of signals saying, I'm full, I'm full, I'm full. But the brain, the CNS neurons, did not have a functional receptor to hear that signal. That's what I'm trying to get you to understand. Now they're in a race to try to make leptin agonists that will try to enhance or simulate the signal that says, I'm full, or Gilrean inhibitors to suppress appetite and help individuals lose weight. And here we have these different points here, which I'd like you to take some time and review. Okay? Now, let's talk about energy for a few minutes. There are different types of heat transfer. We know we have to maintain constant body temperature. We know about heat gain and heat loss must be kept in balance. We are basically creatures that are not, we're not cold-blooded, we're warm-blooded, meaning that our environment doesn't tell us our cues of metabolic order. We have our own means of generating our own metabolism and our own means of generating our own body temperature. But we do not live isolated from our environment. Our environment might be very cold, very warm. We might add to the body temperature by activity. How do we get rid of excess body heat? How do we keep from losing body heat in the situation of, let's say, extreme cold? I put on a jacket. That doesn't help sometimes. But let's go over some of this and I'll show you. Radiation is the loss of heat energy from the body via infrared energy. We all give this off. And think about it. You know, they have those night vision cameras. What are they picking up? Infrared energy from individuals in the dark. Now, evaporation, that's the loss of heat via conversion of liquid water to water vapor. This is due to high specific heat of water. What I'm basically saying is it takes so many calories to convert 1 gram of water as liquid to 1 gram of water vapor. Now, there's two types of perspiration. Insensible perspiration, it's relatively constant at rest. You make a little bit of it. The sensible, meaning that you're going to detect it, perspiration, has a cooling effect when high body temperature. When you have a high body temperature, and this can be due to exercise or high environmental temperatures. Convection is heat loss to the cooler air that moves across the skin. Think about it this way. You move, you've been outside, it's hot, it's 101 out, and you're like, oh, I can't wait. You go into a place of air conditioning, you're like, oh, that feels so good. And what's going on? You've moved to an area where the heat loss from your body can be greatly enhanced because you moved into air conditioning, let's say it's 68 degrees and it's a modest 40% humidity. Boy, is it feeling so much better. The heat is convecting off of your skin quite quickly to the cooler air. Now, conduction is direct transfer of energy through physical contact, okay? I want you to review the physiological effects of failure to control body temperature chart. Here is the different types: radiation, evaporation, convection, that flowing of air over the person is cooling them off, taking some of the heat away, and then, of course, conduction. But if you take a look here, we have to keep in mind some very important issues, not only hypothermia, but hyperthermia. We have fever, of course, that's used to defend against pathogen invasions, sicknesses. We have our normal range, which is 98.6. And we have the lower temperatures, which can, at times, help us to deal when we're very active, etc. But here's the main point. Going too high can have physiological effects. You can end up with denaturing of proteins, convulsions, cell damage, death. If the CNS damage is too great because of things like heat stroke, okay? You can also do it the opposite way, having hypothermia, and you can end up with loss of consciousness, disorientation, and eventually leading to cardiac arrest and death, okay? Now, the thermoregulatory centers in the hypothalamus adjust heat loss and heat gain. Yep. Now I'm going to tell you to review the countercurrent exchange handout. That is in the week, I believe it's week 10 content. Print it out, review it, because you're going to see it coming back again in the renal as well as in several other areas of the course. So it's a helpful tool to review, but I'll show you that in a minute. When we talk about heat loss and heat gain, the control centers are located in the preoptic area of the hypothalamus. Now, what do we do to deal with heat loss? We may have behavioral changes. If we have a high body temperature, what do? Start taking off clothing. You don't have a jacket on, all this other stuff. What also happens? You have vasodilation. You have the shunting of blood to the skin surface. If you have it way too much and you don't have sweat production, this is where you're going into heat stroke or worse. The worst situation you can have is somebody who has stopped sweating, they're red in the face, and they're basically there with you. The stumbles, the bumbles, whatever. Usually sweat production is one of the major survival techniques, okay? And you do also have some loss through your respiration system of heat. And you can see all of these occurring here. Think you don't have a lot of heat loss during respiration? Go out on a nice cold morning and remember what you do. You breathe out and you see this fog. That fog of your breath contains moisture. And when the moisture hits the cold air, it starts to condense a little. But your air that you're releasing out is still warmer than the surrounding environment. Now, you notice a couple of other things here. You're going to have radiation and convection here. This is on the venous flow going back, okay? So you're going to get rid of some of this. Usually what happens is this. When you're talking about getting rid of heat, you're going to have blood shunted more toward the skin to allow for either radiation or convection, okay? Now the opposite occurs if you're in very low temperatures. And I believe I have that in the next section. But let me first go over these few points. In a low temperature, low body temperature or heat loss situation, what you're going to have to deal with is the generation of body heat. You have the non-shivering thermogenesis and then the shivering thermogenesis. Everybody knows what the shivering one is. The non-shivering is involving the endocrine system kicking in to turn up the metabolic activity, okay? The shivering is basically, I'm shivering, it's so cold out, etc. Yes, but remember what that is. That is violent, rapid muscular contractions that generate heat. They're not there to do any skeletal muscle movement work or anything else. We also have the conservation of body heat, okay? First off, you have the vasomotor center decreasing the blood flow to the skin. That's why you start showing up with pale color, bluish color, etc. The lips start getting pale. Certain areas of the skin may start turning slightly bluish. You're pulling away blood from the surface to retain the body heat that exists. You will also kick in the countercurrent exchange. That's why I wanted you to get that countercurrent paper. And the countercurrent exchange is basically you're shunting arterial blood heat to the venous return blood. Remember that there is the superficial venous blood vessels and then the deep ones. Why do we have two? Simple. The superficial ones are great when it comes to getting rid of heat in a hot day. The deep ones, those are going to be taking heat from the arterial blood and returning it back to the heat core. In other words, arms and legs, that's not really as important. Body core temperature, protecting the brain, protecting the heart, the vital organs in the core, is absolutely essential. Now, the interesting thing about this also is this comes into the matter of our countercurrent exchange system. And basically what you have is as the arterial blood flows against these deep veins, the deep veins will take up the heat from the arterial blood flow going out to the limbs. And we tend to conserve our loss of body heat. That's why also, if you notice, when someone gets a real serious situation of hypothermia, it's not, let's say, the core that's going to suffer as much as it is the limbs, and even more so, fingers, toes. When somebody says that I had frostbite so bad that I lost a couple of toes, they meant it because those toes were the areas getting the least amount of heat, and as a result, they would drop in temperature to the point where they would stop having blood flow that would be effective enough to keep the tissue alive. Hence, it would start dying because of lack of oxygen, etc. Okay? Now, you can see this clearly here. Okay? Conservation of body heat, you're going to go from superficial to now deep venous flow, and you're going to have the warm blood from the trunk exchanged over to warm up basically the blood coming back toward the trunk on the venous flow. So this heat transfer is occurring all this time. Okay. Now, before I do that, I want to just hold off here for 1 second. I want to come back here for 1 second, but I want you to see this document. This document is present. I encourage you to read it because when you have countercurrent systems, they can be used for the transfer of a fluid, whether we're talking water, air, blood, etc., nutrients, etc., such as what occurs in the kidney, but they can also be used for energy, such as in the sense of heat. This is a parallel current where basically both of them are following along, and you eventually end up with kind of a happy medium right here. But that's not efficient. If you have one fluid going in one direction and a receiver fluid going in another, you're going to have efficient transfer all the way through. I give you a couple of examples of this, as well as some examples and suggested articles if you would like, but I really encourage you to take the time to review all of this and print this paper out. It's going to be helpful when we start dealing with some of the other aspects of the human physiology. So as we come to the end here, I encourage you to peruse the chapter review outline, pages 927 to 930. Complete the review questions from 930 to 931. Complete the review sheets that you will find on pages 902, 920, and 926. And we are going to move into chapter 24, the urinary system. Have a wonderful day.